Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. In this video, we are going to discuss the structure of nephron. This is the afferent arteriole. And this one is efferent arteriole. The arteriole which is entering the Bowman's capsule, this structure is Bowman's capsule, is known as afferent arteriole, while the arteriole which is leaving the Bowman's capsule is known as efferent arteriole. This is wider. It is having diameter up to 50 micron, and this is narrower, having diameter up to 25 micron. Both are forming a tuft of capillaries, that is glomerulus in the Bowman's capsule. This is a tuft of capillaries. Now this is the Bowman's capsule. It is having podocytes in the inner layer, while the flat cells are present in the outer layer. So here this is glomerulus and this is lining of glomerulus or capillary. So endothelium of capillaries is here, and there is basement membrane below it, and this is the inner layer of Bowman's capsule, which is made up of foot process of podocytes. These cells are podocytes which are forming the inner layer of Bowman's capsule or visceral layer of Bowman's capsule. All three structures, that is endothelium of capillary, basement membrane and inner layer of Bowman's capsule, form the filtration barrier. Then this is the parietal layer of Bowman's capsule, which is made up of flat cells. Then this structure is known as PCT. This is PCT, that is proximal convoluted tubule. It is the major site for selective reabsorption. The cells are cuboidal with microvilli or brush border epithelia. Gap junctions are found in between the cells, and it is made up of two regions: pars convoluta, which is 70%, the coiled part, and pars recta, that is straight part, which is 30 centimeter. The length of PCT is 15 millimeter. Now the next structure is this U-shaped structure, which is known as loop of Henle. So this is loop of Henle. Loop of Henle is responsible for 15% of selective reabsorption and counter current mechanism. This loop of Henle is having length of 14 to 26 millimeter. It depends upon the type of nephron, whether the nephron is cortical or juxtamedullary. Then this is descending limb of loop of Henle. This is ascending limb, descending, ascending. And this is the thin segment of loop of Henle, and this is the thick segment. Now we are comparing the thin segment and the thick segment of loop of Henle. So this is thick segment of loop of Henle, and this is thin segment of loop of Henle. Now we will compare these two segments. If we take the diameter, then diameter is 30 micron. Here the diameter is 15 micron. The cells are cuboidal type. Here the cells are flat. Then the membrane is apical or basal. The apical membrane show less infoldings, while the basal membrane show more infoldings. In the case of thin segment, the cells are flat, so both membranes are flat without infoldings. And if the number of mitochondria is concerned, then it is high in case of thick segment and low in case of thin segment. So the cells of thin segment have flat cells with flat apical and basal membrane, and the number of mitochondria is very few. This is the cell. While this cell is having less number of infoldings in the apical membrane, more infoldings in the basal membrane, and the number of mitochondria is high. It is important to note that in cortical nephrons, the descending limb is made up of thin segment and the ascending limb is made up of thick segment. Descending of thin, ascending of thick. While in case of juxtamedullary nephrons, descending is made up of thin segment while ascending is made up of thin plus thick segment. Now the next structure is distal convoluted tubule, whose length is 5 millimeter. It is relatively impermeable to water, and the nephric filtrate becomes dilute in it. When it passes through the DCT, it becomes dilute. Now the next structure is this, that is collecting duct. So a collecting duct is having three parts, that is cortical part, outer medullary part and inner medullary part. So this is collecting duct. The length of collecting duct is 20 centimeter, and it is having two types of cells, P cells and I cells. The P cells are principal cells and I is intercalated cells. Now the P cells or the principal cells are more in number and their main function is sodium exchange or transport and ADH influenced H2O absorption. While the interstitial cells are less in number and their function is bicarbonate exchange and acid secretion. One another important point to note that the thick segment is meant for active exchange of sodium and water, while thin segment is meant for passive exchange of sodium and water. So this is about the structure of nephron. Hit the like button, share and subscribe our channel to get the latest updates and notifications.